Eledon Pharmaceuticals, Jeffrey Bornstein, and the title is AT-1501 is safe and well tolerated and reduces inflammation in patients with ALS.
Thank you very much. Good morning. Thank you for having me here today. I'm Jeff Bornstein, the Chief Medical Officer at Eledon Pharmaceuticals. And it is my pleasure to present our trial, Tegoprubart AT-1501 is safe and well tolerated and reduces inflammation in patients with ALS on behalf of the study investigators who are listed on the slide, as you can see. I think this is self-evident in an industry talk. So as everybody knows, ALS is a devastating progressive neurodegenerative disease with a heterogeneous rate of disease progression and survival. Increasingly, we are becoming aware of the role the immune system plays in the pathophysiology of the disease. The initial injury is likely brought about by protein misfolding. Then axonal damage occurs, leading to a macrophage response with phagocytosis of Schwann cells and neurons, causing demyelination and T cell activation and infiltration. This in turn leads to microglial activation with secretion of pro-inflammatory cytokines, astrocyte activation, and further neurotoxicity, promulgating a cycle of progressive damage. Tegoprubart, also known as AT-1501, is a monoclonal antibody that binds to Cd40 ligand and blocks its ability to interact with its receptor. This prevents T cell activation via antigen-presenting cells and also B cell activation via T cells. It also promotes T cell polarization to regulatory T cell phenotypes. Animal data in SOD1 knockout mice suggests that inhibition of Cd40 ligand results in decreased inflammatory infiltrate and improved motor neuron survival. We then evaluated Tegoprubart in a phase 2A study in ALS patients. The study was an open label, multiple ascending dose level study. We enrolled 4 sequential dose cohorts, a 1 milligram per kilogram cohort, a 2 milligram per kilogram cohort, 4 milligram per kilogram cohort, and an 8 milligram per kilogram cohort. The lower 2 dose cohorts had 9 participants each, and the higher 2 dose cohorts had 18 participants each. Everybody received Tegoprubart infused every 2 weeks by IV infusion over 1 hour for a total of 6 infusions. To qualify for the study, patients had to have ALS as possible, laboratory-supported probable or definite as defined by the revised ALS score criteria. They also needed to have an ALS FRSR score of 35 or greater at screening and had to be within 24 months of their diagnosis. Presence of a tracheostomy was an exclusion criteria. The primary endpoint of the study was safety and tolerability. We also assessed the PK and anti-drug antibody levels with Tegoprubart. Important secondary endpoints were all biomarker based and focused on target engagement as measured by CXCL13 and Cd40 ligand, and also changes in pro-inflammatory biomarkers. Among the many assessed were TNF alpha, IL-6, enraged, MCP1, IL-1 beta, and C-reactive protein. The study was small and short in duration, but we did look at ALS FRSR and neurofilament light chain. We planned to look at respiratory function, but enrolling the study participants during COVID-19 actually altered the plan on assessing that. So to dive into the results that we're seeing, we'll begin with the demographics. A total of 54 participants were included in the study. The mean age was 59 years of age. 72% were below 65. 2/3 of the population were male, and an overwhelming majority was Caucasian. The mean BMI was 26. The ALS FRSR score at screening, the mean score was 39.9. It did not change substantially between screening and baseline, where the mean was 39.5. There were 4 participants who experienced a significant drop between screening and baseline. And there were 6 other participants, all in the higher 2 dose cohorts, who had significant bulbar impairment at baseline. We screened 70 participants to enroll 54. 15 screen failed. One participant who actually enrolled in the trial was replaced before their first infusion. So we dosed 54 total. 52 of them had perfect compliance with the protocol. Only 2 had significant protocol violations. 91% of the study population completed all 6 infusions and the follow-up visit. And among the 5 who failed to complete, all 5 had a minimum of 4 infusions, 1 having all 6 infusions, just failing to show up for their end of study visit. 2 of the participants were discontinued due to adverse event. 1 in the lowest dose cohort, the 1 milligram per kilogram cohort, discontinued after the 4th infusion due to depression. Another participant in the 2 milligram per kilogram cohort discontinued also after the 4th infusion due to malaise. As noted, 1 participant in the 2 milligram per kilogram group completed all 6 infusions, but did not return for their end of study visit. And 2 patients in the highest dose cohort withdrew due to not wanting to return every 2 weeks for further infusions after their 4th infusion. In neither case was that associated with an adverse event or with a significant change in their ALSFRS score. In terms of safety, a total of 217 events were reported in the 54 participants of the trial. 78% of participants had at least 1 treatment emergent adverse event. There was only 1 serious adverse event in the trial that was deemed not related by the investigator. As noted, there was 2 events leading to withdrawal, and there were no deaths in this trial. Looking at the most frequent reported adverse events, they were fairly much in line with what would be expected in this population. The most frequently occurring AEs were fatigue, fall, headache, muscle spasms, and you can see the list there. Nothing changed our view of the safety of Tegoprubart after this trial, and importantly, no thromboembolic complications were noted. Shifting to our secondary endpoint now, focusing on our biomarker assessments, we'll start with the biomarkers of target engagement. We used 2 different biomarkers. CXCL13 is more of an indirect measure of target engagement. It's a marker of B cell activation, and as noted previously, the mechanism of Tegoprubart should block B cell activation. What we saw was a nice dose response in terms of mean reduction in circulating CXCL13 levels, plateauing at the 4 milligram per kilogram dose. The numbers at the top of the slide represent the percentage of participants in each cohort who had at least a 10% decline from baseline in CXCL13, 10% being chosen to overcome the variability of the assay. And you can see that 17 out of 18 participants in both the higher 2 dose cohorts achieved this. Notably, the effect was seen early on in the treatment course and was sustained throughout the treatment period. A more direct measure of target engagement is looking at Cd40 ligand itself. And you can see that, once again, the 2 higher dose cohorts, the 4 milligram and 8 milligram dose cohorts, achieved greater than 50% reductions in Cd40 ligand levels, with almost all of the participants experiencing at least a 10% decline in that level. Once again, the effect occurred early and continued throughout the dosing period. Moving now to biomarkers of pro-inflammatory activity, another secondary endpoint in this trial. We used a chip that had 96 biomarkers or 96 different proteins on it, of which 36 were relevant to inflammation. 20 of the 36 had a statistically significant reduction from baseline at one of the 2 higher dose cohorts. And shown here on the slide are the 10 that had the greatest reduction from baseline. And you can see, similar to what we saw with the target engagement, we had a dose response where more biomarkers moved more at 2 than at 1, and then it plateaued at the 4 milligram per kilogram dose cohort, where 4 and 8 were really producing very similar type of effects. I should note, not shown here is neurofilament light chain, which is not a biomarker of inflammation, but is a relevant biomarker in this disease. It did not change with Tegoprubart therapy, although higher levels at screening did correlate with a more rapid progression on ALS FRSR. Also not shown is the ALS FRSR scores themselves, which, as mentioned earlier, in a small trial of short duration, it did not look different than what is expected from historical data. Notably, in these biomarkers, the reductions seen occurred in biomarkers that demonstrate reduced T cell activation, things like TNF alpha and the interleukins, reduced B cell activation, IgA, IgE, and IgM, and reduced chemotaxis seen in CXCL9 and CXCL10. So to summarize, our trial demonstrated that in this patient population, at these doses, Tegoprubart was safe and well tolerated. We demonstrated target engagement in a dose-dependent fashion. Proof of mechanism of inhibition of inflammation, also seen in a target-dependent fashion, affecting both T cell and B cell activation. We conclude by saying that we believe further study is warranted to assess how this might translate into clinical benefit in this patient population. Thank you for your attention. I'm happy to answer any questions you might have.
Thank you so much for this. That is, it's wonderful to see this work advancing so well after all the years that have gone into the development of this particular product and reagent. Thank you for that. I had a couple of questions. One is, have you looked for immunoreactivity to the product itself, to the antibody?
So we looked at anti-drug antibodies, and I didn't mention it, actually, I should have. We had a very low rate of ADAs in this study. We've done in vitro work on whether this might paradoxically produce a hyperreactive response, and we did not see any increased signal. And of course, we didn't see any in our clinical trial or our other clinical trials that are ongoing in other indications.
So no T cell reactivity to the product. Right. Yeah, that's excellent. Thank you. And can you say a little bit about how you want to go forward with this? And in particular, is there particular merit in substratifying the cohorts with different ALS gene mutations, which may be more or less predisposed to inflammatory pathology?
Yeah, thank you. I think that's an excellent idea. We've certainly tried to look at our existing data set to get as much out of it as we can to inform our next study. Our next study will have to be much larger. We'll likely have to be dose ranging, and will have to be long enough and with enough participants enrolled to truly assess clinical outcome. Stratifying by things like genetic mutation or neurofilament light chain or disease severity or time from diagnosis are all important considerations, things that we're looking at as we currently plan the next trial.
Wonderful. Wonderful. Thank you again. That's an excellent and much appreciated presentation. I think at this point,